Hi everybody, welcome to Clinical Biomarkers. This is our very first lecture in this subject. Now I know I'm listed as the instructor of record, but a lot of this class design is here to feature Dr. Beltran. Caroline Beltran has been a postdoc at Stellenbosch University for just under 2 years?
3 years.
3 years now.
It's going on 3 years.
She arrived even before I did, my goodness. And she is an expert in biomarkers. She spends all of her time thinking about this subject, and I was really, really grateful that she was willing to work with us to create for the first time a course in biomarkers. So I'm thrilled that you have all decided to take a chance on us, and I think it will be worth your while. Now she's going to introduce herself a little bit, but I would point out that the structure of this class should be reasonably familiar to you. We're going to have Tuesday and Thursday lectures at 10 o'clock. One exception, however, is next Thursday. Instead of having a lecture on Thursday the 9th, which is a holiday, we're going to slide that lecture one day later to Friday the 10th. So Friday the 10th, we will have a lecture at 10 o'clock. Otherwise, it's Tuesday, Thursday, 10 o'clock. We're going to have, 4 lectures from Caroline to begin with. I could remember those topics if I were probably not flashing right now, but we've got the Introduction to Clinical Biomarkers. We have the immunoassay lecture, that's Thursday. We have a lecture on proteomics, biomarkers, and then we'll have a quick talk about transcriptomic and genomic biomarkers. After that, we go a little bit quantitative. I'm going to be back up in front of you guys on the 14th and 16th of August. We'll be talking about receiver operator characteristic curves and introduction to machine learning, which I'm really excited about. It's my first time giving a lecture in that space. And then she'll be back for one more Tuesday when she talks about case studies in clinical biomarkers. So this is going to be a lot of fun for us, and I hope it's going to be a lot of fun for you as well. We are going to stick with an assessment strategy for this module that is ongoing. So the material you see today will appear on a quiz at the start of class on Thursday. The material you get a lecture on Thursday will appear on a quiz the following Tuesday. So everyone should be aware of that. The final quiz will take place on the case study day. Everyone ready for that?
Yeah.
All right. If you are auditing the class as a ringer like our lecturer, Dr. Ashwell back there, Ashwell Klein back there, then you should be aware that you will also take the quiz. Great. All right. We will not, however, read out your grade aloud and laugh. No, just kidding. Okay, so does everyone have the broad structure of how this works? There's no formal assessment paper that we all take at the end. Your grade is summed across all of these quizzes. They're multiple choice questions. Be aware, I write tough multiple choice questions.
He does.
Some of the students were like, "You're asking what?" It's going to be a lot of fun. So I think you're all going to do really well, and I look forward to seeing how that turns out. All right, with that, I'm going to take the video camera and turn the mic over.
Right, hi everyone. I'm really happy to be able to teach you just for the next couple of weeks. As Dave says, this is a bit of a work in progress, so there might be glitches here and there, but I hope by the end of this you'll have a better understanding about how to study biomarkers, what you should take into considerations if you're going to do a big study to look for new novel biomarkers, and basically there are a lot of things you should take into account, and I hope you're going to touch on that. This is by no means a very comprehensive biomarkers lecture. We could probably spend the whole year talking about biomarker design, studies, case studies, and things like that, but hopefully we'll get enough knowledge out of this that you guys have a better understanding of biomarkers and clinical research. I think most of you, from what Dave told me, have done plant biotechnology and are more into like the wine biotech, is that right? Or a lot of you worked in clinical research already? No? No? Okay. So most of this will be on human studies, of course, but any of this can be applied to animals or plants or any other system that you're wanting to investigate. So I want to tell you guys a little bit about myself first and where I came from. So originally I studied my undergrad and my honours at UKZN. I actually did a lot of plant pathology back then and looking at the kind of diseases that affect plants. I then came to UCT to do my master's and my PhD, and I did it in abalone. So I don't know how many of you actually have seen an abalone, but this is an abalone here under the ocean, and this is what they look like when they've been shucked so that they can be eaten. And they're really interesting creatures, and I didn't really know that much about them when I first started. But on farms where we farm all over the Western Cape, there are actually quite a lot of diseases that affect abalone. But one of the things of abalone is they don't really show any clinical signs of disease or any kind of symptoms. So what ends up happening is that diseases spread on the farms without the farmers really knowing about it. Hi guys! Without the farmers realizing what's happening, and all of a sudden you just get mass die-offs on these farms. And they really had no way of measuring how these animals were responding to infections. So part of my PhD was to do an immune challenge on these animals. And I worked at the aquarium down at Sea Point, which is, I don't know if you've ever seen this big grey building right by that Sea Point beach. There's an aquarium there where we study like a whole bunch of animals, and I used a quantitative whole proteomic strategy. So I did eye track for the proteomic experts in the audience. I did an eye track investigation, just basically characterizing the proteome of these abalone when they were infected with mock infections as well, and then just obviously a normal uninfected control, and we looked for biomarkers. I went on to validate these biomarkers, and they're actually starting to test some of these biomarkers out in the farms. So I've inadvertently been studying biomarkers for quite a long time, and now, as Dave mentioned, I'm at Stellenbosch. I work in the immunology research group, and we're looking at tuberculosis. And my specific interest is biomarkers of treatment response. A lot of our patients, I think some of you will know TB treatment is extremely long. It takes a really long time to cure a patient, and often we're not sure when a patient has reached true sterilization. So I've been working on different things like imaging and different techniques for staining the bacteria and growing them up in different types of medium to try and see if we can find these last little bits of persisters that seem to be in patients at the end of treatment, but we can't identify them. So that's really where I come from. Oh, I want to disclaim I am a notoriously fast speaker. I'm trying to work on it, but if you need me to slow down or repeat something, please just put up your hand and I will try and slow down. It's my curse and I'm working on it. Also, I really like that we're in this kind of format because I think we can have a really nice discussion, so please don't hesitate to put up your hand, stop me if you want to talk about something, and let's just discuss everything we're going to go on. So today is basically an introduction to what we're going to be working on. So I'm going to just talk about the types of biomarkers that we have. So the learning objectives here, we're going to define what a biomarker is. Talk about what characteristics of a good biomarker are, what do we want in a biomarker that we're developing for the clinic or for the wine industry. We'll talk about different biomarker types because there are actually quite a varied classifications. We'll talk about anatomical molecular biomarkers. We'll talk about why panel biomarkers are really becoming more ideal and what we actually want in the field. We'll talk about the discovery pipeline of biomarkers, which is really complicated and fraught with lots of disasters and pitfalls. And then we'll talk about what can go wrong in your biomarker discovery. So what is a biomarker? Why are we interested in biomarkers? Does anyone want to tell me what they think a biomarker is? Don't be shy. Right, yeah? You want to go for it? No? Okay, so biomarker is short for biological marker. In our bodies there is, and I'm just talking about humans, but obviously we can apply this to any organism, there are various molecules and anatomical features that can be indicative of either being healthy or having some sort of disease. And this is not only for looking for diseases, but you can also use biomarkers to identify when someone is responding to a treatment or is having some sort of effect due to a drug. So really common biomarkers that we would use, say like fever for, a flu virus, or high blood pressure, or your glucose level when you have diabetes. Like these are all biomarkers. But we're starting to move into really measuring large amount of biomarkers to try and really tease out what's happening in diseases and get something that's highly specific for a specific state. So there are various broad definitions, and there's this NIH Biomarkers Definition Working Group that is trying to standardize how we talk about biomarkers and what exactly do they mean. So a biomarker, according to the NIH Biomarkers Definitions Working Group, is any characteristic that is objectively measured. So that's very important. A biomarker, you should be able to quantitatively measure this characteristic. So any characteristic objectively measured and evaluated as an indicator of either normal biological processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. Ideally, you want your biomarker to change as a patient's responding to treatment or as he's getting better and have an indication of how the disease progresses. Also, what's ideal is if your biomarker can tell you if a patient's having an adverse reaction to treatment. So biomarkers can really encompass a whole host of different biological responses that are happening. For another definition, this is from the WHO, it's any substance, structure, or process that can be measured in the body or its products and influence or predict the incidence or outcome of disease. So ideally, your biomarker should be clinically useful. And what I mean by that is it's all very well to take a biopsy from inside the body and you find this protein that's highly expressed, but you can't find it anywhere else. If it can't help, at the end of the day, the end point of making a decision of what's happening with that patient or that organism, then it's not going to be clinically useful. So you want a biomarker that's going to be easily measurable and really indicate a specific disease. So I tried to just summarize it with, it's a measurable biological component that can be analyzed and indicates a specific disease state. But this can also be normal if the patient goes back to normal processes. So what are the characteristics of a good biomarker? Like what, actually maybe let's talk about that before we go through it. What do you guys think is a good biomarker? What makes a good biomarker? We already talked about it must be easily measurable. Yeah? Quantifiable, good. Yeah, you need to be able to quantify it because often just presence or absence is not good enough. Being able to measure it, having a tool that can measure it is really, really important. What else?
Should it be sensitive and specific?
Yes. Seems like you've done this before. Sensitive and specific. That is so important and that's what you will always hear. And I think Dave has maybe something about sensitivity and specificity. But you want your biomarker to be unique, hopefully, to your state or your condition. It needs to be highly specific because if you're measuring it, but it indicates a whole bunch of different processes, it's not going to be very useful, especially if you've got quite a complex scenario you're trying to deal with. It also needs to be sensitive. If you need a highly specialized mass spec to measure your protein of interest and it's not going to be rolled out into the clinics, that's going to be very hard to implement as a good biomarker. So sensitivity is also very important. So let's talk about other things. So specific to disease, we've already talked about that. High sensitivity, safe and easy to measure. I think most biomarkers are trying to work either blood, because you can do a finger prick test, or urine. Urine is like the gold standard of biomarkers, but also if anyone's worked with urine in mass spec, they'll know how difficult it is to work with urine. But something that you can easily get from a patient without causing them undue pain is an ideal biomarker. It needs to be rapid for fast diagnosis. If say your protein, I keep talking about proteins, I'm sorry, but if your protein comes up only much later when the patient's got a really terrible disease, well, it's not going to be very useful. You ideally want something that comes up nice and early and definitively diagnoses your patient quickly into treatment. Cheap, that's always what the companies are going to go for at the end of it. Is it cheap? Can we manufacture this in a cheap way? So usually you work with a large scale of lots of molecules and you start refining down to these biomarkers. But if you can't develop a test like a lateral flow test, and we'll talk about that next lecture, it's going to be very hard for any company to kind of go for your biomarker. Consistent, that's also important. We get a lot of interpatient variability and intrapatient variability, and depending on health status, what they're eating, where they come from, you're going to have all of this variability, and maybe your biomarker doesn't come up. Maybe it came up in your population that you were studying, but it's not going to be for everybody. So it needs to be a consistent biomarker. And able to differentiate different states, this is the ideal goal of a biomarker, but often quite difficult. So if it can tell you if you've got someone who's progressing maybe worse or actually responding to treatment, or someone is in early stages of disease about to develop, say, tuberculosis, that's really an ideal candidate. So all of these characteristics of a good biomarker is what we all aim for, but a lot of this you end up not being able to do. And you have to make concessions in your biomarker and know where its limitations are. But this should always be playing in your back of your mind if you want to roll this out, or if you actually want to apply this in the field, do they meet these standards? Other things we can think of is significantly increased in the disease condition. That's probably the best scenario you have. Maybe if your protein disappears when you have a disease, it's much harder to measure the absence of something. So ideally you want something that comes up in your disease and you can measure it very easily and sensitively. If it can measure the effects of treatment intervention, that's also gold standard. Economical, quick and consistent, and then accurate and reproducible, quantifiable. Right. Everyone okay with good characteristics? And I'm sure you can think of many more other good characteristics. This is not an exhaustive list. So there are a number of different types of biomarkers depending on what they're showing or what they are being used for in, say, the clinic. So the first type of biomarker is called a predisposition biomarker. So this is also known as a risk or a genetic susceptibility biomarker. Most of the time, a predisposition biomarker is genetic, most of the time, not always. And if you have this marker, it means that you're at a higher risk of developing the condition. It doesn't necessarily mean you will, but if you have this genetic marker, usually you may have a higher risk, and then a lot of the time we use predisposition biomarkers to screen patients and put them into an arm where maybe we'll do an intervention early on. We monitor these patients to see if they're going to go on to develop the disease. So that is your disease risk susceptibility biomarker. And we're going to go into real life examples straight afterwards so we can get a good idea of what this means. Diagnostic biomarkers, most of us end up working on diagnostics. They're also called trait biomarkers. So your diagnostic is, do you have the condition? Do you have the disease? So they confirm the presence of the disease or your disorder. And ideally, as I spoke about, you want it to be early detection. That's really the key. I think some of you will know, for example, the quantiferon or the TST test you do for TB. That actually doesn't measure if you have active TB because also latently infected people have it. So they're always trying to work on something that will actually identify patients that have active TB or an active disease case. Okay, so diagnostic biomarkers, pretty self-explanatory. Prognostic biomarkers, if you think about in the hospitals when they say what's the prognosis for this patient, right? This predicts the risk or outcome of a disease. So how, based on these biomarkers, is this patient going to progress? Maybe he has good prognosis and his tumor is not so enlarged and he's actually got a good prognosis and is going to respond well. Or it's not looking very good, it's stage 4 cancer and he has a really bad prognosis. So prognosis biomarkers are really important. And they're usually based on intrinsic factors based on the patient themselves. So that's usually what you take into consideration and you will measure to make a prognosis. Prognostic biomarkers also measure the likelihood of a clinical event, so what's going to happen to this patient in the course of the disease, but also whether or not they're going to have a recurrence or relapse, right? Maybe they'll be cured, but because they're not showing very good, say, a PET-CT score, they might actually end up relapsing and getting the disease again. Or however, we've talked about the progression of the patient through the disease. And this is different, the prognostic biomarkers are different to predictive biomarkers. So predictive biomarkers is when you have a therapeutic intervention. So based on the patient and these biomarkers, how will the effect of a treatment affect the outcome of this patient? How, if I treat this patient with this specific drug, will this patient respond to it? So it's different to prognostic, where here you're just looking at the disease, how does this person look? Okay, he looks like he's going to progress this way. If I give him this treatment based on his biomarkers, he's going to have a good outcome. So there's a very slight, small difference, but it's quite substantial. So here, usually with predictive biomarkers, you want to try and stratify your patients. So identify those patients that are probably not going to respond to your treatment, push them into the arm of non-responders, and maybe try a different intervention. So it's really important. And usually with biomarkers, you want to have this whole suite to complement the disease that you're studying. But this is really hard, obviously, to meet all of these requirements. Finally, treatment response biomarkers, very similar to predictive, but slight difference. This is how the patient is responding. So how you've now given him this drug, is he responding to your therapeutic intervention? Or is the tumor getting smaller? Is his chest X-ray resolving? Does he still have activity when you're doing a PET-CT scan? That kind of thing. So is your treatment working for this patient? So if we look at actual examples, so I took breast cancer. I think most of us know about breast cancer. So in terms of a predisposition biomarker for breast cancer, have most of you heard about BCRA genes, BCRA 1 and 2? So if you have a mutation in either of these genes, you are at higher risk for developing breast cancer. It doesn't necessarily mean you will develop. So basically, if you've got a mutation on BRCA1, you've got about 55 to 65% risk of developing breast cancer. On BRCA2, 45, and then normal BRCA, you still have a risk for developing the disease. But here you can make a decision for your patient if they have these mutations, maybe have a mastectomy or something like that. In terms of diagnostic biomarkers, usually a tissue biopsy is taken to get a definitive diagnosis, or we can do a mammogram, and there are now a lot of protein blood-based tests. Cancer biomarkers is probably the biggest source of knowledge if you want to read about biomarkers. It's active area of research, lots of money being poured into cancer biomarkers. For prognostic biomarkers, usually tumor size. They look at the tumor, whether it's what kind of stage. You can go stage 1, stage 2, stage 3, stage 4. And if you're getting, if it's spreading to the lymph nodes. So usually if it's spread to the lymph nodes, you have a bad prognosis for your cancer. In predictive biomarkers, something that's used in breast cancer is the presence of estrogen and progesterone receptors. Based on the amount of these receptors that you have, you will benefit from a hormone treatment. Whereas patients that don't have this kind of profile need a different type of chemotherapy. So based on this, if your patient has estrogen and progesterone receptors, lots of them, you would then choose this kind of therapy for them. So that's an example of a predictive biomarker. And then finally, treatment response biomarkers, most of them are dealing with imaging. So PET-CT, it's where you inject with like a radioactive compound, and you can measure the amount of glucose activity in the lymph nodes or on the tumor. And based on that, you're looking for the change in tumor size. Hopefully it's decreasing, hopefully you have less activity. And based on that, you can say, okay, chemotherapy is working, we can carry on with this treatment or change the treatment. Okay, so that's real life examples and the types of biomarkers that you have. Is everyone fine with that? Does anyone have any questions? So there are two basic type of properties, and I realize I have not covered anything about pharmacological biomarkers because that's a much more complex area, and we could probably do a whole lecture on that if you really, really want it. So I'm talking about here mostly physiological biomarkers or molecular biomarkers, which we'll go into more details in the other lectures. So physiological or anatomical biomarkers are basically any functional process in the body that you can measure. So blood flow in the brain after a stroke is a type of biomarker that we use to see if you've got treatment success. And they're usually based on imaging techniques. So like I mentioned, PET-CT activity or MRI scans to look at the brain activity. These are all anatomical biomarkers. Things like your blood pressure, anything that your body really you can measure in the body of a patient is going to be your anatomical biomarker. And typically, usually, these kind of biomarkers are non-invasive, which is ideal. The problem with, can anyone see the problem with anatomical biomarkers? If you're just measuring processes in the body, usually they're not that specific. There are a lot of very specific ones, especially if you're studying a tumor and you're measuring the tumor, like the tumor's there and you can see it. But a lot of things like high blood pressure could be indicative of lots of different things. So that's the problem with anatomical biomarkers, but often they're used in conjunction with other type of biomarkers. So molecular biomarkers are the ones we're really going to be focusing on for the next couple of weeks. And there are a whole, you guys must know the central dogma, right, with genes being transcribed, mRNA, and then we get proteins, and then we can get lipidomics and all of that. So we can start with genomic biomarkers, and we talked about these are usually risk biomarkers, not always. So for example, the BRCA genes and breast cancer. Transcriptomic biomarkers, usually they're looking for these multi-gene molecular fingerprints. That's what they like to call fingerprints for breast cancer survival. Based on your signature, based on the amount of transcripts and how they're expressed, usually ratios of one to the other, you can generate quite a very powerful signature for that kind of biomarker study. Proteomic biomarkers, probably my favorite area of research. So proteomic signature, especially now with the advances we're making in mass specs and our detection tools, proteins are really powerful to look at. And that will be next week's lecture. We'll talk about why proteins are really an ideal source of biomarkers. Also sometimes not. Metabolomic biomarkers, we're not really going to touch on that, but just keep it in mind. All the metabolites that happen, often with a disease, you get metabolites that are being excreted in the urine that are really easy to measure. You can use mass spec, for example, develop a lateral flow test. So metabolomics is starting to become really, really big field in biomarker studies. Sorry. Cytometrics is also an area for flow cytometry. I don't know if anyone's done flow cytometry at this point, maybe for your projects. Pardon? Yeah. Cell cycle analysis is definitely something you can look at. The types of circulating cells that you have, are your T cells differentiated? Anything like that, where you're measuring how the cells in the body are responding, or what they look, or what type of cells you have, is cytometrics and is a type of molecular biomarker. Everyone good on this? So let's talk a little bit about single versus panel biomarkers. So originally, single biomarkers are based on kind of older methodologies, right? When we didn't have like the best tools to study everything. We would look for a single protein or a single microRNA. And these are quite laborious to screen because if you're screening one at a time, it's going to take ages to find something that's really shows you something ideal. And usually when you're looking at single biomarkers, they're based on pre-existing knowledge. So you'll have been studying this, you'll have done functional work into how this disease progresses, and you're like, okay, well, in this pathway, I think maybe that this is up. And so you'll start looking for this target and start measuring it in a whole bunch of patients and see if it's up. So it's quite a simple study, and it's low cost. If you're measuring a single biomarker, you don't really have to develop very intricate ways to screen it. But it's quite variable, and it's really not specific. So a really good example is C-reactive protein. Have you guys heard of CRP before? Some people are saying yes. So CRP is a protein that is increased 1,000-fold in the blood when you have inflammation. So very easily detectable. It comes up very strongly, very quickly. And so they've used it to diagnose a bunch of conditions based on other knowledge. The problem with CRP is it comes up for viral infections, in cancer, in diabetes, sepsis, even smoking, cardiovascular diseases. So unless you're a clinician that really knows the history of a patient or what's been happening, it's going to be very hard to say, well, CRP is up, it must be cardiovascular, or it must be diabetes. So although it's a really good biomarker in that it's 1,000-fold increased, you can detect it, it's easy, it really isn't very specific, and that's the problem. So when you have variability of disease or variability of diseases, a heterogeneous population, these biomarkers become very lowly accurate, not very specific and not very sensitive for what you're trying to identify. So single biomarkers, they have their use, certainly when you get to a point where, for example, pregnancy tests are measured on a lot of biomarkers, those are very simple biomarkers, but you need to make sure it's very specific. So everyone really, especially in biomarker development and studies, we're moving to panel biomarkers, so multi-signatures. We've entered now, I'm sure you guys are always hearing, we're in the omics era, right? Like transcriptomics, proteomics, metabolomics. We're able to now measure thousands and thousands of molecules very quickly, very accurately, if you're designed your study properly. And we have also the computing power that's able to take this entire mess, all of these proteins or all of these transcriptomic signatures you have, and make sense out of it. So we're really starting to move. If you're going to work in biomarker development, you will probably work on a multi-panel signature. And of course, if you have multiple signatures, you're going to increase your specificity, right? Measuring, say, 100 compounds together is going to be much more specific than just a single one, unless you know that one is only for your specific disease. So we talk about multiplex. Multiplex signatures are multiple markers that are measured simultaneously. So you will do a transcriptomic signature where you'll take this patient's blood and measure every single transcript that she has, and then put this into a signature and see what's up or what's down, or how the transcripts correlate with one another. This is different to bundled biomarker signatures. So that's where you have independent assays, and you're taking a bunch of different types of biomarkers and using it to make a decision about the disease. So it could be blood pressure with the presence of a protein with another anatomical feature, and you'll use that as a bundled biomarker signature. So as we've said, these multiplex signatures are much more sensitive, much higher specificity, and you get very large quantitative information for a large cohort. Because if you can measure in a single, say, mass spec run, thousands of proteins for one patient, and you do everybody, you can do that pretty quickly. Well, maybe not on a mass spec, but you can do this for quite large cohorts quite nice and quickly, or using microarrays. And so this is moving towards this thing that I think you might have heard, like personalized medicine. So your signature is different to yours, and so we're going to tailor the treatment for you specifically because of your biomarker signature. But of course, this is much, much, much more complex. The data outputs that we're getting now are huge. We're generating, what was it? We've generated more data in the last 5 years or 10 years than we have in, I don't know how many hundreds of years. So we're entering this era of large data sets, and you need to try and tease this out and actually understand what's going on. So a lot of the problems with these studies when they're done is either the study design is partially flawed, maybe you've been a bit biased with who you've selected, and then once you start rolling out these biomarker signatures that don't hold out, or you haven't done the analysis correctly, or the data is just too complex. You also need to be able to normalize this data across everybody, and that becomes quite difficult. So these multi-panel signatures really have quite a lot of problems attached to them, and hopefully as we do the next lectures, we'll talk about where they can go wrong, what you need to think about when you're doing the data analysis, and hopefully Dave also covers that in intensive detail. So multi-panel signatures, much more specific, much more sensitive, and really the goal of your biomarkers. Okay, so in the biomarker discovery pipeline, I actually had like 20 slides about this, and Dave told me to just narrow it down. Usually when we start a biomarker discovery, so say you want to do the study and you're like, I want to study this prostate cancer, and what are the different gene signatures of patients who have prostate cancer? You're going to do, like, let's say a transcriptomic, like an RNA-seq data set, and you're going to look at all the RNA, all the mRNA's transcripts, and you're going to start in your discovery cohort with a very large amount of candidates. Possible candidates, if this is your early exploration, you've just started and you've got a very small cohort, maybe 10, 20 patients, maybe a little bit more if you're lucky, if you've got enough money for that, and you're just doing an early exploration to see if there's anything here that's worth investigating. So you're going to come out with your list and you're going to show your supervisor, like I have found 200 upregulated proteins or mRNAs that look like they might be important. So here you've got a large number of molecules and maybe a very small sample set. And you're going to have to move down this pipeline to identify which ones are going to hold out. At each level of the pipeline, you're throwing out biomarkers. This one doesn't come up in everybody, tossing it out. I can't reproducibly measure this one, I'm tossing it out. Along your pipeline, you toss out biomarkers that just don't fit your criteria anymore. So you'll move on from discovery to confirmation. So are these candidates viable? So here you'll start increasing your sample size. You've got lower candidates that you're measuring, increasing your sample size slowly. Are these viable? Can I measure them in, say, urine or the blood, or are they only specific to an organ? You then move on to, you started now selecting certain candidates, you want to move on to validate these candidates. So your validation state is usually when you're optimizing your assay. So now you're going to do an SRM strategy. You're going to see if you can consistently measure this very nice and specifically sensitively. So usually you characterize at the validation stage, you'll characterize the specificity and sensitivity of your candidates. So on your pool here, where you started up with lots of marbles, you're starting to funnel down. We're at validation here. Usually you're sitting at around maybe 10, 20 possible candidates that you are pretty confident about. You've really confirmed them. They came up really nicely in all the first stages. You've chucked out everybody else that's really not meeting the standards that you want. And you're starting to optimize your assay. And usually at the validation stage is when you start having multiple time points. So maybe if you've got patients that you're following longitudinally, you're going to see how does this biomarker change with time. Does it stay stable? Does it change if the patient's getting better? Does it disappear? Are they not always measurable at different time points? So you're really trying to tease out exactly how this biomarker behaves in your population. So here, you're having much more subjects in your pool here. Finally, you've now come up with this list, let's say 5, and you're like, these biomarkers are perfect, they're great, and they really accurately diagnose my patients quickly. So you're going to write a grant and hopefully get funded to do a clinical evaluation. So usually phase 1, phase 2, phase 3 clinical trials happen for biomarkers, and you're going to evaluate it in a real-life clinical scenario. So here you're looking at very, very high sample size, usually in the thousands of patients. So you're trying to see the variability between patients. Is it being affected here? Does my biomarker still hold out? You've got a lot less control now over your cohort. You've got a lot less control over how sample, because of course there's a lot of sampling issues that happen right in the clinic. When you're in charge of your study, you know how it was collected, you know how it was processed, but in real life scenario it doesn't work that way. So does your biomarker still hold up here? Does your signature still hold out? So usually you've got a high sample size and 10 analytes. So as you can see, the discovery pipeline just starts funneling down into just a few candidates. So you start out with 1,000 that you were super confident about. At the end of it, you've got 5. But 5 is good enough if it can diagnose your patient or if it can monitor treatment response. So always keep that in the back of your mind. You're going to go down in the biomarker pipeline discovery. and you want high confidence candidates. So we're just going to end off, this is actually quite a short lecture, I don't know how long I've been talking for, about the pipeline discovery pitfalls. So biomarker discovery has really been something everyone's on top of, everyone wants to talk about, everyone wants to study biomarkers, but although we see papers that come out with excellent signatures, it looks good, it's holding out, for some reason they never make it out into the field. Like they just disappear. And there's the discovery to implementation failure is exceedingly high for biomarkers. Most of them fail. Most of them will never make it to any sort of clinical trial. And that's because biomarker discovery, as I'm sure you're starting to realize, and probably have realized with your own studies, is really difficult. You know, it's really hard to get something that's specific and sensitive and is going to hold out and be super strong throughout your study. It's very expensive to look for biomarkers. As I'm sure you know, kits and all that, especially if you're doing early exploration work, it's expensive. And also as you move down clinical trials, you have to look at thousands of patients. It gets more and more expensive. And companies maybe don't want to fund these kind of studies. Very time consuming. These can take years to get to the point where you've actually even got a signature. Decades of research and of hard work. And often funders don't want to carry on funding these kind of studies. And there's a lot of confounding factors in the biomarker discovery. If you think about it, we're looking at patients that come from all over the place, that have different genetic backgrounds, that eat differently, maybe some smoke. You're not dealing with the perfect scenario where you're controlling a cell line, or even you're studying mouse models where they're all genetically identical. You've got so many confounding variables that are going to throw spanners into your biomarker discovery pipeline like you won't believe. Tissue localization, some biomarkers are only very specific to a certain area, the site of disease. So maybe when you were studying it from a functional point of view, something comes up in the lungs and you can detect it. Maybe you were looking at cadavers, for example, and you managed to get brain tissue samples. And that biomarker is there and it's in the brain. But how are you going to measure that in patients who are alive? You can't. So often that happens, you find something that's good, but it just is not present. What's that?
Spinal tap.
Yeah, spinal tap, we can do that, that's true. But the patients don't want to go for a spinal tap. Yeah. Persistence of the biomarkers. A lot of these biomarkers seem to come up and down. They're just not consistently up for whatever reason in the blood or in the urine, or someone drank too much Red Bull that day, so now you can't measure it. So this failure to identify these factors, that's why you need to always think about what can go wrong. What is it about my population that I've studied that may be not reproducible in another scenario? We've done biomarker studies across Ghana, Uganda, and South Africa, where the biomarker signature is completely different. You would think this transcriptomic signature is going to hold out in TB patients, but it doesn't. It's different. Ghana patients have a completely different transcriptomic signature. So for some reason now we cannot apply this biosignature to different countries. So that's really problematic. You can't really, you can work on a biomarker specifically for your country, but ideally you would like to roll it out. So also another problem is bias. And we kind of talked about that. So we talked about if you're going to study a biomarker retrospectively, in the past, you had all these patients that were sampled for whatever reason. You're going to take all those blood, all those saliva samples, and you're going to look for a biomarker. The choosing of those patients was biased. You're not looking at like everyone in the population. You're like, oh, well, these ones look like they might have the disease, so I'm going to study in them. So of course it comes up strongly in them, but it's not going to be that prevalent in a mixed population, if you will. Does that make sense? So you have a bias in your study, which is inevitable at times. You have to choose which patients you're going to look at, and that bias can really cause problems downstream. Because although it came up in your pool of 10 or in your pool of 20, as soon as you're doing 1,000 patients, you see nothing. And you're just wondering why. What did I do this whole time? And now nothing is showing up. So you must always be aware of the biases in your study design and try and fix them early on. Try and make sure that you are studying something or you are choosing patients that are representative. Now, of course, in the beginning, you do want patients have the disease, of course. Otherwise, it's going to be impossible. You need to screen thousands of patients. But always have that in the back of your mind that you could have this bias that might become problematic later on. So just to recap what we talked about, and this is going to be helpful for the quiz. A biomarker is a measurable characteristic which indicates a biological state or a condition Remember, it doesn't necessarily have to be a disease. It can be a normal process or a pharmacological response. An ideal biomarker should be specific to your condition, sensitive, easily measurable, and able to differentiate different states of your disease or your condition. Biomarkers can be classified based on their application, whether they're prognostic or predictive or diagnostic. There are different applications for a biomarker, so you should classify it accordingly and decide what do you want to work on. Do you want to find a diagnostic biomarker or treatment response? There are different considerations for a type of biomarker you want to work on. These can be either anatomical or molecular, and we've talked about the differences between both of them, which I'm sure, and we'll go into molecular diagnostic development and how you should go about it. Panels offer improved sensitivity and specificity over single biomarkers. So a panel is going to be more specific, but it will take more computing power and more understanding of your condition to tease out properly. Finally, a biomarker discovery pipeline begins with many candidates, and it will end with a few candidates that you will roll out. And it is fraught with pitfalls and problems, which will end up in you not getting funding at the end of it. Yeah, so that's it for the introduction to biomarkers. Do you guys have any questions for me? Or any comments? Or any of your own experiences with biomarkers? When I was doing the iTrack research on the abalone, I found about 30 that were really significantly upregulated in my disease. And then as soon as I started studying it over time, I managed to culture abalone hemocytes, which are their blood cells. And as I measured over time, I realized this biomarker really comes up within 30 minutes of the bacterial challenge, and then it just completely disappeared. So then it's like, is this biomarker going to be useful in a farm where disease could have been happening here, here, or here? So in the beginning, I was really excited because it came up super strong, and I was like, oh, this comes up super early, this is great. But in fact, if it's just going to disappear after an hour, or it's very short-lived, or it has a short half-life, it just becomes useless. So I had to chuck that one out, which was my favorite, and work on a different set of candidates, which weren't, in my opinion, doing as well, but actually they're coming up quite nicely on the farm. So it's tough. It's a tough business to be in, basically. Cool. Thanks, guys.